Hello, everyone. Thank you for having me at the Second World Congress on Endocrinology, Diabetes, and Metabolism. My name is Jonathan Snyder, a PhD candidate in the Sato Lab at Drexel University College of Medicine. And today I'm here to tell you about our work investigating the role of GRK2, that's G protein coupled receptor 2 kinase, in pancreatic islets and in insulin secretion. I'll start off by telling you all a little bit about why we hypothesize that GRK2 might play an important role in insulin secretion and in islet biology. This largely relates to GPCR regulation and insulin secretion. And then I'll tell you all about two important inroads that we've made into investigating that hypothesis. The first was our work in GRK2 knockdown model in the Min6 model for beta cells, and we've investigated the role of GRK2 in energetics and insulin secretion there. And then after that, we also developed a pancreatic specific GRK2 knockout mouse model where we've so far investigated glucose handling, body composition, and insulin secretion. And then I'll conclude by sharing just a little bit of our data going forward and what we think this all means in the schemes of type 2 diabetes and islet health. So canonically, GRK2 is known for phosphorylating ligand-bound G protein coupled receptors that signals for beta-arrestin recruitment and internalization, and that terminates the signaling of that specific G protein coupled receptor in most cases. So in the absence of GRK2, which is observed in the DB/DB mouse pancreas, not quite the absence, about 50% abundance compared to a control mouse, you could hypothesize that the absence of this internalization, this well-regulated internalization process, would result in a hyperstimulation of G protein coupled receptors in the presence of a ligand because they would not be internalized. One of the challenges in investigating the role of GRK2 in any specific tissue is that it does this for quite a number of important receptors, some of which are ubiquitously expressed. For instance, a couple of receptors that I've been thinking about that are expressed in the islet that GRK2 could be regulating to modify insulin secretion include opioid receptors, cytokine receptors, specifically the adrenergic receptors, and different metabolite receptors such as the free fatty acid receptors. Furthermore, I do just want to give the caveat that GRK2 is a fairly promiscuous kinase, and it has a complex interactome outside of G protein coupled receptors. And finally, one of the big drivers of our interest in GRK2 is that its inhibition has been specifically noted for potential therapeutic utility in mouse models with heart failure and therefore in patients with heart failure. This has been investigated both through small molecule inhibition and also via peptide inhibition using a gene therapy such that the inhibition would be specific and limited to the heart. It is important to note that GRK2 is upregulated in heart failure, and we're only talking about bringing its activity back down to normal, healthy physiological levels. One other really important note is that global GRK2 inhibition has also been shown to improve glucose tolerance in the DB/DB mouse. However, it's sort of the opposite situation as in heart failure, because in the DB/DB mouse, the GRK2 is already downregulated in the pancreatic islets and pancreas, which I had mentioned earlier. So it might not have that the effect that it's having globally may not be as much due to an effect that's already happened in the islet specifically, and that paper did show that at least part of the effect of GRK2 inhibition was due to improved insulin sensitivity. So speaking of insulin sensitivity, many of you here may be familiar with this graph in particular and broadly with the model of clinical progression for type 2 diabetes, where initially insulin secretion and insulin resistance increase both in tandem. So the body becomes less sensitive to insulin, but the islets are capable of responding to that by secreting more insulin, such that normal glycemia is maintained both in a fasting state and in a post-meal state. However, that doesn't continue forever, and the islets do begin to, specifically the beta cells, begin to de-differentiate and to die off to a certain extent, resulting in a deficit of insulin secretion relative to insulin sensitivity. What's really interesting when it comes to G protein-coupled receptor regulation is that G-alpha-S stimulation, such as with the GLP-1 receptor agonist, which has been very successful in the treatment of diabetes, not only improve acute insulin secretion, but they are also found to improve islet beta cell differentiation and overall islet health. On the other hand, G-alpha I coupled receptors, such as the alpha-2 adrenergic receptor, not only acutely impair insulin secretion, but also diminish the capacity of the beta cells to expand their population and improve long-term capacity for insulin secretion. So before I get into the studies that we've done here, I just want to remind everyone of the canonical model of insulin secretion and a couple of the factors that can amplify or impair that canonical model. First of all, glucose enters through non-insulin dependent glucose transporters. It undergoes normal metabolism to produce ATP. That ATP, in a concentration dependent way, inhibits the ATP sensitive potassium channel, potentially resulting in the depolarization of the cell and the opening of voltage-gated calcium channels. That influx of calcium results in the efflux of insulin-containing granules into the bloodstream. This pathway can be modified in many ways through other metabolites that contribute to oxidative phosphorylation, through G protein-coupled receptors that modify oxidative phosphorylation, or intracellular calcium stores. And you can see that GRK2 could play a role in either through its canonical regulation of G protein-coupled receptors. Our lab has also been specifically interested and has published on GRK2 interacting directly with the mitochondria to modify substrate preference and oxidative metabolism. So now moving into some of our recent work, we started off, like I mentioned, in the beta cell line Min6, and we investigated different points along the canonical pathway of insulin secretion, here starting with mitochondrial efficiency. So what we did was we simply knocked down GRK2 expression using an siRNA. And what we found was that we were able to knock it down to about 50%, which is similar to that which is seen in the DB mouse pancreas. We are showing the knockdown here both by qPCR as well as by Western blot, and we did check that our knockdown was specific to the GRK2 isotype and that we were not intervening on any of the other GRK isoforms. We then used seahorse respirometry to measure oxygen consumption in a few different conditions at baseline, and we found that GRK2 knockdown had no influence on baseline oxygen consumption. We then treated with glucose and saw an approximately equal increase in oxygen consumption in all cell groups, showing that glucose is entering the cells effectively and stimulating respiratory metabolism. We then treated with oligomycin, which shows us how much of that oxygen consumption that was just previously happening was dependent on driving ATP synthase. Oligomycin is an ATP synthase inhibitor. And what you can see there is that the cells that had GRK2 knockdown continued to consume more oxygen, showing that the oxygen that they were consuming was not necessarily tied to efficient production of ATP. We then treated with FCCP, which is an ionophore, and we can measure absolute maximal metabolism. And finally, rotenone and antimycin, which inhibits the entire electron transport chain, and we can measure non-mitochondrial oxygen consumption. You can see that there are no differences there. We quantified this in two important ways. One, which is coupling efficiency, how much of the oxygen is driving ATP synthase. And you can see that there is a significant decrease in coupling efficiency. And that was in part due to proton leak, so how much oxygen the mitochondria are consuming that's not going to ATP production after subtracting out that non-mitochondrial fraction of oxygen consumption. And showed that the knockdown resulted in increased proton leak. Importantly, we also used a separate assay where we very simply measured the ATP concentration in cell lysates using a luciferase-based system. And what we found was that both at low glucose, approximately 3 millimolar glucose conditions, the knockdown resulted in less ATP, and also at high glucose, maybe 16.7 millimolar, glucose, thinking post-meal levels of glucose, there was also less ATP in the cell lysate there. Thinking back to the canonical model of insulin secretion, if there's less ATP being produced, you would expect less depolarization of the beta cells and less influx of calcium. So we also measured calcium dynamics using a calcium-sensitive fluorescent indicator. We kept the cells at baseline at 5 millimolar glucose for a few minutes, then stimulated with glucose, 25 millimolar glucose, and found that there was, again, approximately 50% decrease in this calcium peak occurring after glucose stimulation in the cells that had GRK2 knocked down, shown in red here. What's really interesting here is that we introduced an inhibitor of the G-alpha IO subunit of the heterotrimeric G protein coupled receptors. So now we're inhibiting the inhibitory G protein coupled receptor subset that we believe GRK2 might interact with. And what we found was that this rescued the effect of GRK2 knockdown, showing that GRK2's inhibition, the inhibition of calcium signaling induced by GRK2 knockdown was dependent on effective G-alpha IO coupled signaling. So finally, all of this leads to insulin secretion events. So we repeated that same paradigm of low glucose and high glucose treatments, and also the pertussis toxin inhibition of G-alpha IO. And what we found was that indeed, like we had seen with the diminished ATP production, diminished calcium flux, there was also diminished insulin secretion in vitro with these Min6 cells in the Jesus assay, glucose stimulated insulin secretion. However, when we treated with pertussis toxin and blocked G-alpha IO signaling, we were able to rescue this effect once again, showing that GRK2 is potentially interacting with a specific subset of G-alpha IO coupled G protein coupled receptors. So what have we shown so far here? Well, we've shown that it appears that glucose enters the cells just fine. We showed that with the seahorse data. And based on the extracellular acidification rate, which I did not show you, it appears that at least glycolysis remains intact. But oxidative phosphorylation has been diminished. ATP production has been diminished. Calcium flux has been diminished, and insulin secretion. So it appears that wherever GRK2 interacts with this system, it is downstream of glycolysis. In another study that I'm not showing here, we also showed that intracellular calcium release appears to remain intact. So it is not a simple matter of G protein coupled receptors influencing intracellular calcium release. It is more likely an interaction of GRK2 with GPCRs that then interacts with oxidative phosphorylation, or potentially a direct interaction of GRK2 with oxidative phosphorylation components. And I'm just sort of showing our model of that here, where GRK2 might be interacting directly with the mitochondria, like we've shown in previous studies in our lab in different tissues, not yet in the islets, or that GRK2 is interacting in this pathway via G-alpha IO coupled receptors. So like I mentioned at the inquest of this presentation, we then developed a pancreatic specific knockout of GRK2. Here you can see an exemplar Western blot where GRK2 is knocked out fairly efficiently in the pancreas and to a high degree of efficiency in isolated islets. And we did a simple OGTT in these animals where we gave a bolus of glucose, normalized their body weight, and found that the animals that had GRK2 knocked out in the pancreas had a great deal more glucose exposure, and that this was due to a deficit in insulin secretion shortly after that glucose introduction. And I do want to highlight that this isn't in the absence of any traditional research diabetes challenge, such as a streptozotocin injection or a high-fat, high-sucrose Western type diet. We did some fairly simple immunohistochemistry on these islets for insulin and glucagon and found that the islets were of similar size and similar cell type distribution. However, we did interestingly find that there was more fluorescent signal upon insulin staining from the knockout islets, suggesting that there may be providing further evidence that this is a deficit in insulin secretion rather than in production or in cell type differentiation. So to take this one step further, we did end up doing a high-fat diet, 60% of their calories from diet in these mice. We found that they gained more weight over an 8-week period, interesting, while eating less food. We also found that these animals had an even more exacerbated glucose exposure during OGTT, which was once again due to a deficiency in insulin secretion. So what can we conclude from these studies? Well, from a molecular standpoint, we can say that beta cell GRK2 expression is important for supporting efficient metabolism, calcium signaling, and insulin secretion. Furthermore, it appears that pancreatic GRK2 in mice is critical to glucose management via insulin secretion. We do need to do further studies, and we have initiated further studies in being sure that this is specific to the beta cells and that it is not an artifact of that mouse model that I showed earlier, being from the embryonic stage forward. So getting more to the clinical relevance of this project, GRK2 inhibition is of great preclinical interest for heart failure. It has shown efficacy in different mouse models of heart failure. And some clinical trials have investigated whether paroxetine in SSRI that happens to inhibit GRK2 could provide some utility in heart failure. And I think what our study goes to show is that if you're working with a patient who has both heart failure and is either type 2 diabetes or is pre-diabetic or for some other reason predisposed to diabetes, that should be approached with caution, because you might be not only inhibiting their ability to secrete insulin in an acute capacity, this could also be intervening on their long-term islet health. Second to that, like I mentioned, paroxetine, which is used which is an SSRI, but it has also been investigated for a protective role in heart failure. What role could using paroxetine as an SSRI play in metabolic health? There was one study that showed that it did lead to more adverse metabolic outcomes compared to other SSRIs, but I think a meta-analysis that looked at a larger group of patients showed that it was not a major effect. So finally, I think what this project speaks most to is the importance of GPCR regulation in islet development, performance, and health. The success of GLP-1 receptor agonists in type 2 diabetes management also lends credence to the importance of this fact. However, I don't think we have a strong understanding of what's going on the other end of this, the G-alpha-I inhibitory input to the islet. How much is that playing a role in ultimately islet failure to secrete insulin to the extent that an individual's insulin resistance might demand? So going forward with this, we'd like to determine whether the role of GRK2 that we've shown in the pancreatic GRK2 knockout model is specific to beta cells. To do that, we've developed a cell-specific and doxycycline-inducible GRK2 knockout model for both alpha and beta cells. And then we'd also like to investigate whether GRK2's effect on insulin secretion is primarily associated with its canonical GPCR regulation role or one of its many alternative functions that have also gained a lot of interest and research traction. Specifically, we are interested in GRK2's translocation to the mitochondria and any role that it might play in respiratory efficiency or substrate preference. I just want to give you a little taste of data that we have in the beta cell specific inducible knockout model. Here's a Western blot showing that we're knocking GRK2 down in isolated islets to about 20% of the control, which is about commensurate with 80 to 90% of the cell population in the mouse islet being beta cells. And what we found so far is that after 12 weeks on a high-fat, high-sucrose diet, that these animals do have modestly more glucose exposure, but a fairly significant insulin secretion deficit shown on the right there. So it may be that there's both a time-dependent, diet-dependent role for GRK2 in islet biology. So thank you guys so much for listening. I'd like to thank the different members of the Sato lab, past and present, especially my mentor, Dr. Sato. And thank you to the department and the great individuals that support this research from the department, and of course our funding, and especially the good people at the Penn Institute for Diabetes, Obesity, and Metabolism. Thank you.